Throughout days 0 through 4, the fertilized egg takes a journey through the uterus as it undergoes a series of changes. During day 0, fertilization occurs when the chromosomes combine with the egg to form the diploid zygote, which is a fertilized egg. This becomes the first cell of the newly developing individual. These chromosomes then replicate as soon as they come together. Throughout day 2, cleavage takes place. This is a period of rapidly growing meiotic divisions without intervening growth. The first cleavage division results in 2 blastomeres. These then divide to produce 4 cells. On day 3, the morula forms, which is a berry-shaped cluster of at least 16 blastomeres. By day 4, the embryo consists of 100 cells. The embryo begins accumulating fluid within an internal cavity and floats free of the uterus. The zona pellucida begins to break down, and the inner structure, now called the blastocyst, hatches from it.
Beginning with the chorion, which is seen on the outermost part of the membrane, this contributes to the formation of the placenta. On the bottom is the maternal portion of the placenta, as well as the fetal portion of the placenta. Next comes the amnion, or amniotic sac. This forms when the cells of the epiblast fashion themselves into a transparent membranous sac. The embryo itself is seen, as well as coming off of the embryo, the yolk sac, which forms from the cells of the primitive gut and serves as an early site for the formation of blood. Yolk sac is pictured. Next is the allantois, formed by an outer growth of the embryo's gut, primarily involved in nutrition and excretion, and is webbed with blood vessels. It collects liquid waste from the embryo and aids in gas exchange, seen here. And of course, the middle portion between the allantois and the yolk sac is the umbilical cord.
During week 3, the 2-layered embryonic discs will transform into the 3-layered embryo. During this stage, the 3 germ layers will form and the extra embryonic membranes will develop. Before becoming 3-layered, the inner cell mass subdivides into 2 layers, the upper epiblast and the lower hypoblast, which together are called the embryonic disc. During a phase called gastrulation, the epiblast will form into the ectoderm, and the hypoblast will become the endoderm, and the mesoderm will begin to develop between the two. The ectoderm, mesoderm, and the endoderm are collectively called the primary germ layers, which will serve as the primitive tissues which all body organs derive from. The ectoderm will become both the central and peripheral nervous systems, cells of the adrenal medulla, the epidermis of the skin, hair, glands of the skin, and nails. The mesoderm will become the dermis of the skin, connective tissue, heart, muscles, skeletal system, kidneys, gonads, adrenal cortex, and the blood vessels. The endoderm will become the epithelial lining and glands of the digestive and respiratory tracts, the liver, the gallbladder, pancreas, the thymus, thyroid, and parathyroid glands.
3 weeks after fertilization, the embryo forms the foundation of the heart. By 6 weeks, the heart begins beating on its own. The fetus has specific modifications to the circulatory system to compensate for the underdeveloped lungs in the womb. All of these modifications are altered at birth once the baby starts to breathe. The ductus arteriosus is a shunt that connects the pulmonary artery and aorta so that blood can enter the systemic circuit and not enter the underdeveloped pulmonary circuit. Once born, the ductus arteriosus closes to form the ligamentum arteriosum. The foramen ovale is a hole between the 2 atria, which helps the oxygenated blood from the umbilical veins bypass and travel more efficiently to the systemic circuit. After birth, this hole becomes a depression in the atrial wall called the fossa ovalis. Another shunt in the developing embryo is the ductus venosus, which allows blood from the placenta to be shunted into the inferior vena cava, bypassing the liver. This shunt forms the ligamentum venosum once the fetus is born. The fetus receives oxygenated blood from the umbilical vein, which carries blood through the ductus venosus in the liver. This structure later formed the ligamentum teres. The deoxygenated blood is carried away from the heart through two umbilical arteries to the placenta. These arteries form the medial umbilical ligaments once the fetus is born.